Good morning, good afternoon, good evening to all that are joining from all across the world. I am Florent Ginoux from the Singapore Immunology Network from A-Star. And it's my pleasure and honor today to host and introduce our Global Immunotalk speaker today, Professor Gabrielle Belz. Dear Gabrielle, could you share your video? We can see you. Hello, nice to see you, Gabrielle.
Hi.
So before introducing that, just a reminder that we will have Dr. Mazopust next week on June 9. And so please do join us also on June 9. So today we have Professor Gabrielle Belz from the University of Queensland Diamantina Institute presenting live today from Australia. And the recorded session will be replayed later at the regular Global Immunotalk time. So Gabrielle Belz is an Australian molecular immunologist and viral immunologist. She was born in Brisbane, Queensland, Australia, and trained in veterinary medicine and surgery at the University of Queensland. And she has made major contribution to the field of immunology, for which she received a Doctor of Veterinary Science. So Gabrielle completed her undergraduate studies at the University of Queensland, graduating with a Bachelor of Veterinary Biology in 1990, then a Bachelor of Veterinary Science with first-class honors in 1993. She completed a PhD in 1997 at the University of Queensland, and following her PhD, she took up a postdoctoral fellowship in viral immunology with the Nobel laureate Professor Peter Doherty at St. Jude's Children's Research Hospital in Memphis, USA. So she returned to Australia to take up a position with the Walter and Eliza Hall Institute, the WIHI of Medical Research in 2000. And after a significant time in the WIHI, she moved back to Queensland to take the chair of immunology at the University of Queensland Diamantina Institute. So her prime research interests are in the area of infectious disease, particularly in the lung and gut disease, where she worked to unravel how protective innate and adaptive immune cells are wired to generate long-lived protective memory. More recently, she has become interested in unraveling how innate cells can protect against tumor, and she continued to be excited by the unexpected and new discovery linking innate cells in dynamic physiological regulatory circuits in immune protection. So we're about to start. I just want to precise that Gabrielle has published over 200 peer-reviewed original papers in leading scientific journals, but also have received multiple awards for her accomplishments, including in 2007, the Burnett Prize, which is recognizing the Australian virologist Sir Macfarlane Burnett. And she opened the prize for her research into how dendritic cells and cytotoxic T cells work together to defend the body from viral infection. She was also awarded the Gottschalk Medal by the Australian Academy of Science for her contribution of a series of pioneering discovery, illuminating how the immune system deals with virus. And she also was awarded an NHRC Elizabeth Blackburn Fellowship Award. And very importantly, in 2018, she was elected Fellow of the Australian Academy of Health and Medical Science, which is a great achievement. So again, Gabrielle, thanks so much for accepting our invitation and for giving a Global Immunotalk entitled A Game Changer, Innate Lymphoid Cells, Eosinophil Collaboration Reshapes Melanoma Outcomes. But before I ask you to share your slide, Gabrielle, you will not, there is a tradition that we have to have a question to the speaker. And the question for you today will be, who are the scientists that have inspired you the most and why? Gabrielle, please.
Oh, that's a great question. And I think with COVID, we see so many great scientists stepping up to the mark. But I have to say the two people that have inspired me the most are my PhD supervisor, who is probably not widely known, who worked on lymphatics in the body and really taught me how to do really strong science. He didn't work in a really sexy area at the time, although it's become much more prominent now. But I think that really gives you the basis of doing good science. And then I think Peter Doherty, not because he got a Nobel Prize though, but because of his infectious love of discovery. And I think if people can hold on to that throughout their career, they're in good stead to really be fascinated by the problems that come along that we still need to solve.
Thank you very much for this response. And I think now it's time for you to share your slide with us, and you can start your presentation. And I will unmute and remove the video. Thank you very much again.
Okay, so can everyone see that well?
I think it's working. I can see them, so.
Thanks very much. Thanks for that wonderful introduction and for the invitation to actually talk today. I think we're all sort of in remote positions, but we still have this opportunity to connect with each other. What I really wanted to tell you about today is a story that we've been developing over the last 4 or 5 years, and now we've finally rounded this up. So this will be published in another week or 2 weeks, but hopefully I can give you a little bit of an insight into some parts of the story and understanding how innate lymphoid cells reshape the outcomes of melanoma. And so I have to preface this by saying that this is really driven by a very talented postdoc who was in my lab, Nicholas Jacquelot, and he really drove this work in a very robust fashion, and has now just moved to Canada to take up a postdoc over there to do some studies in human melanoma. So melanoma is a deadly disease, as you can see from this map, that Australia, United States, and some parts of Europe are the most severely affected by this disease. And in fact, Queensland itself is the melanoma capital of the world. So there's nearly 250,000 cases of new melanoma diagnosed every year, and about 55,000 people die from it annually. And one of the things that's really problematic about melanoma is it doesn't have a great success rate for treatment. So survival was really partitioned into 4 different stages. So localized melanoma, which is stage 1 and 2, regional melanoma, which is stage 3, and metastasized melanoma, which is stage 4. And you can see on this graph that once you reach metastasized melanoma, the chances of surviving, irregardless of intervention, is very, very low. So obviously we've made great strides forward in different types of therapy, and melanoma therapies have been part of that story. You can see here with immunotherapy, we do increase the percentage of people who survive longer term, but overall we're not significantly increasing the chances of people having long-term survival. So after about 5 years, only about 50% of all people who have melanoma are actually still alive. And we are able to treat with immunotherapies, as you can see there, but they also seem to come with some very unpleasant side effects, such as disruption of the gastrointestinal tract, various lung and skin disorders. So the immune system is really a fascinating network of cells. And there are many ways in which we can target the immune system to try and help us deal therapeutically with diseases that we encounter. So in the case of melanoma, possibly identifying predictive biomarkers, identifying mechanisms of therapy resistances and trying to overcome these, try to reduce therapy-associated toxicities, and finally combining different types of therapies to really have an onslaught to try and tackle this sort of serious disease. I think over the past decade, we've really focused on just one aspect of the immune system, and that's the adaptive immune system. And the work that I did in Peter Doherty's lab really focused on these very fancy cytotoxic T cells. They're wonderful cells because they go around killing things that are infected or exhibit features that make them look not normal to other cell types. And this has really taken up a lot of our time in trying to understand the heterogeneity within that adaptive immune system and how we develop immunological memory. And it's this immunological memory that really provides a protection against repeated exposures to pathogens. So more recently though, and I think in a really wonderful way, we've discovered a whole new group of cells that parallel the adaptive immune system. And this has come about by the development of new tools by our group and a vast number of labs around the world that have allowed us to have a little insight into what's really happening in the body, only to discover that cells that might fit into non-conventional cells or innate type cells form an equal number of cells as the adaptive immune system. So it's now really clear that the immune system is much more complex than we previously appreciated. And it's these innate cells that really provide minute-by-minute defences in acting as communication hubs that liaise between the external environment and the adaptive immune system. And our objective in the lab has really been to try and tackle understanding some of these innate cells, and we're really interested in the innate lymphoid cells, and trying to decipher how they come together to work with the adaptive immune system to mediate immune protection. It also importantly really identified a major gap in our fundamental knowledge, because for the last number of decades we've been trying to solve problems by just considering how the adaptive immune system operates without really thinking about are there other elements that could be contributing to this particular network. And so we've treated a lot of problems symptomatically, and understanding this innate arm of the immune system perhaps gives us some capability to develop more complete therapies by understanding the picture more completely. So innate lymphoid cells, one of the reasons I think that they're really interesting is because they're really the frontline protectors. They're often referred to as tissue resident, so mainly they're tissue resident, although not exclusively, but importantly, they're found underlying all the natural barriers of the body. So the skin and the mucosal surfaces, and the mucosal surfaces really cover a vast surface area of the body. They rapidly respond to different types of stimuli because of their anatomical positioning. They're positioned so that they can see pathogens or disruptions or inflammations very, very quickly. And because they don't require specific T or B cell receptors, they can respond to those sorts of stimuli quite rapidly. What's quite perplexing about these cells is that although they can respond very quickly with different sorts of armories such as cytokines, there are a number of situations that they exhibit what we've often referred to as paradoxical responses, and that is they liberate these cytokines in such abundant amounts that they drive autoimmunity. So understanding the balance that regulates whether they produce protection or autoimmunity and pathology is really pivotal. We also know that they have a number of key immunomodulatory properties, and these haven't really been explored very carefully. And as hopefully by the time we get to the end of this talk, you'll understand that probably we should dig a bit deeper on this. So innate lymphoid cells fall into three major groups, and these really parallel the types of groups that we've designated for CD4 T cells. So group 1, ILCs are composed of the prototypical cell, the NK cell, together with non-NK ILC1s. They depend on the transcription factor T-bet, and they produce the cytokine interferon gamma. The group 2 cells, they depend on GATA3, and they produce a number of cytokines, including IL-4, 5, 9, and 13. But 5 and 13 are really the most abundant. And then ILC3s, or the group 3 ILCs, depend on the transcription factor raw gamma T and produce IL-17 and IL-22. So in the context of melanoma, though, we first asked this very, very straightforward question: do innate lymphoid cells infiltrate melanoma tumours? And so this seems like a very simple question, but is slightly more complicated by the rarity of these cells. We wanted to look at this in a system that was a bit more like the human situation. So there are multiple different models for melanoma, many of which depend on injecting cells, and we will use some of those systems. But for most of our experiments, we use a system in which we have a mouse that expresses the two genes, PTEM and BRAF, and we can conditionally inactivate the PTEM, which drives BRAF mutant expression and results in the spontaneous formation of melanomas. So about 20 days after we treat the mice, we start to see the emergence of these melanoma lesions on the animals. These start off quite small. They take much longer than usually an injected cell type to form these melanomas. So when we isolate these cells, and we've isolated the cells from the skin and the tumour and then other organs as well, And then on the left-hand side, you can just see the gating protocol that we use. So ILCs are typically referred to as lineage negative, and this mainly referred to a more historical version or definition in which they don't express T-cell receptor, CD3, CD19. We now know that they do express a number of markers of importance. But we use a whole suite of markers to remove those lineage positive cells. So CD19 here for B cells, CD11B for monocytes. And then we take those residual cells and stain them for a variety of markers that they do express, which includes NKp46, which distinguishes ILC1s and NK cells, and raw gamma T and GATA3 to distinguish ILC2s and ILC3s. And when you do this, you can see that there is a relatively small but consistent number of ILCs that are present in these tumours. And particularly the NK cells and the ILC2s have infiltrated these tumours. These numbers are significantly lower than for T cells, but nevertheless, I think we can't get completely fixated on having huge numbers of cells reflecting that they're meaningful, as we know that there's a very heterogeneous group of cells that are involved in immune responses now. It's worthwhile considering in our models that maybe bigger is not always better, and that we should consider some of these smaller populations that may be, in fact, very influential players. So because Nicholas was very interested in really what happens in human melanomas, we tried to parallel all of our studies with looking at exactly what happens in human melanomas. And so we were able to have a very wonderful collaboration with researchers at the Peter MacCallum Cancer Centre. So Paul Neeson, Minyu Wang and Kevin Tai, who assisted us in looking at primary and metastatic samples to try and determine if we could see ILC2s in these samples. And so through a series of optimization of these different stainings, we were able to use a relatively small number of markers to identify that within our tumours, we do have these ILC2s present. And so here on the right-hand side, you can see the frequencies of those cells for both primary and metastatic melanomas. And you can see that NK cells are reasonably abundant in these, in addition to other ILC subsets. So as I mentioned, these cells are relatively rare. So really what role do they play, or are they just sort of bystanders in this response? And so we were able to address this by using a number of different mutant mice that we had ready access to, and comparing B6 mice with RAG1 deficient mice. RAG1 deficient mice lack B and T cells, and RAG2 gamma C deficient mice, in which they lack B cells, T cells, and the ILC subsets. And in this case, we did intradermal injection of a RET tumour cell line that's derived from melanoma. And so we're able to make these induced melanoma tumours. And if we look at the survival curves and the tumour growth from these treated mice, we find that if you have a B6 mouse, you're relatively long-term protected. However, if you lack T cells and B cells or ILCs, you're very susceptible to developing large tumours fairly quickly. And so what we can see, though, from the RAG2 gamma Cs is that there's a level of protection that's mediated just from the ILCs and a level mediated by the T cells. So the adaptive immune cell system and the innate system are working hand in hand. And this is reflected also in the long-term survival of these animals. So if you lack both adaptive and innate immune cells, you're going to die about 50% more quickly than if you have the full complement of the immune system. So, as I mentioned, NK cells are pretty prominent infiltrate into our tumours. And we know that NK cells are quite important in some of these melanoma models, mainly from the point of view of dissemination, but not in primary tumours. Nevertheless, we checked whether we really wanted to analyse whether NK cells were involved in this process. And so we took advantage of a conditional mouse that was generated a couple of years ago, and that is the NKp46 or NCR-CRE crossed to an MCL1 flox mouse. So in this mouse, they lack NK cells, ILCs, and NKp46 ILC3s. So immediately we're eliminating 3 of the populations that could be playing a role in these tumours. We then injected the mice intradermally with the ret tumour cell line and looked at the tumour growth in those mice. And you can see here how you get this nice population of NK cells in our wild-type mice. And then in our our control mice, you still see this population of cells, whereas in the conditional mouse in which we've expressed the CRE, we now get complete ablation of these cells. If we now look at what happens to the tumour size and the protection in survival, we find that there's not really any difference in the tumour sizes that develop, indicating that that impact of NK cells in this primary tumour development is not so great. Again, in the overall survival, this impact is relatively small and wasn't statistically different. So from these studies, we can concur that the NK or NCR expressing ILC subsets are not playing a role in this particular development of the tumour. So which cells really are playing a role? And so we had an inkling that ILC2s might be playing a role in this development. And we had access through Andrew Mackenzie in the UK, who's provided us with a number of ILC2 models. And the first one that we used was the IL-7 CRE crossed to the raw alpha mouse. And so this mouse lacks ILC2s, but raw alpha is also expressed by other cell types. So there's probably some mild impairment of other cell types in this system, which is worth keeping in mind. However, if we now treat with our tumour cells and look for survival, you can see that there's this nice ablation of the ILC2s in this system. And then we have this very distinct difference in the tumour growth size, and this translated to protection as far as survival goes. So the question is, what are ILC2s doing, and how can we demonstrate in a much more specific way what ILC2s are mediating in this response to the tumour? And so we've used a system which I continue to be perplexed that it actually even works, and that is a reconstitution of RAG2 gamma C mice with very small numbers of innate immune cell progenitors and bone marrow progenitors. And so by knowing the different stages of the bone marrow development into ILCs and adaptive immune cells, we're able to purify each of these subsets, so CLP, ALP, the ILC2s and ILCPs, to generate these different types of cells, transfer them into mildly irradiated mice, and then track the response to challenge with the tumour cell line. So this just shows you the setup, how we identify these different cells for purification. So looking at FLT3 positive cells, and then taking the c-KIT positive cells and separating out the ILC2 and the ALP. We then transfer each of those different types, so the ILC2, the ALP and the CLP, into our mice, wait for 6 weeks, and then inoculate them with the tumour, and then map their capacity to restrict the development of those tumour cells. When we do this, and here we've just shown you the control data essentially that I've shown you previously, and then we combine this with the RAG2s in which we segregated out each of these donor progenitor cells. And what you can see is that if you have CLPs, you reconstitute something that's a bit more akin to B6 immune system. So these cells are able to produce all the different cell types. But intriguingly, if we take ILC2 progenitor alone, we generate almost as much protection or restriction of the growth of the tumour size compared to the ability to produce both the adaptive and the innate immune cells. So this was a very striking finding for us, and it was highly reproducible. As I mentioned, we were very interested in what really happens in patients though, because we're not really trying to cure mice of melanoma. And so we have a really fantastic collaboration with Melissa Davis and Saror, who are bioinformaticians. And we were able to work with them to provide gene signatures of some of our cell types and ask them to model these types of gene signatures based on our data and other people's data by taking tumors from the TCGA database and looking to see if there was a relationship between patient survival and this ILC2 signature. And so what you can see here in the primary tumor with the ILC2 higher cells, we only have a very limited number of patients, but this trend to protection if you have high ILC2 signature. And similarly, even in the metastatic tumors, the more ILC type cells you have, the more likely you are to have a longer survival. So that was very encouraging for us. So the big question though is, now that we've described this very interesting sort of pathway, what would be the molecular pathways that could be involved in this process? And that's where it becomes a fair bit more tricky. As you can see from this single cell t-SNE plots, there are lots of cells that infiltrate a tumor. And so here we've just taken some of our tumors from our BRAF mice, and we've digested them up and done single cell sequencing on the entire tumor. And so there's a whole myriad of cells that are in there. And you can see highlighted in this red circle are the ILC2s. So proportionally, they look like just a very small fraction of the cells that could be contributing to the response. If we then dissect this up, and there's many ways of doing this, but one of the things that we started to work through were some of our cytokines of interest and some of our genes of interest, and look at those and whether they are expressed more specifically by ILC2s compared to other cell types that might be in the tumour. And so you can see here for the ILC2s, they're producing IL-13 and they're producing IL-2, all the cytokines that might be associated with those cells. But they're also producing some genes that we had not really expected, so GM-CSF or CSF2. And we were very interested in this because we were actually working with Eric Vivier at the time, and we were very interested in this notion that ILC3s produce GM-CSF, and that's work that's been described by Miriam Merad. And so we really wondered how the ILC3s worked, only to discover that perhaps ILC2s were also producing GM-CSF. And so when we probed those cells to look to see if they really were doing that, these are cells that are out of the lung and the tumour itself, and really focusing on the ILC2s. Within the tumour, you can see that there's this massive number of cells that are producing the GM-CSF. And in this case, we've tracked them by doing intracellular staining for the cytokine, but we also have a direct reporter that replicated this sort of result and allowed us to look at RNA expression versus protein expression in real time. So this to us was quite a striking finding. What is the GM-CSF actually doing, and does it contribute to this response? And so we were fortunate at the time. We had both the knockout mice and Ian Wicks at the WEHI, together with Warren Alexander, were making a flox mouse, which gave us a number of tools to explore this question. And so if we just take the complete knockout, we find that you can see that there's quite a large difference in the tumour size if we completely lack GM-CSF in our hematopoietic system. We also see a vast difference in the survival of the mice when they lack GM-CSF. And if we go back to our TCGA database and look at our samples, and here I'm just showing you metastatic samples now, and ask if we tandemize having a high GM-CSF and high ILC enrichment, does this affect patient survival? And here you can see that if we have a high CSF expression, CSF2, we actually have a really increased potential for increased survival and better outcomes for the patients. So how do the ILC2s actually mediate this response? And what is the impact on the tumour? And so the first thing we did was go back to our adoptive transfer system and ask what sort of cells might be affected by the progenitors that we put into the mice. And we looked at a vast number of cells, and I've just put a few of the different subsets up here. So dendritic cells, monocytes, macrophages, neutrophils, and eosinophils. we know that ILC2s play an important role in recruiting eosinophils, principally through their role of producing IL-5. Separately, we had looked at IL-5 knockouts and IL-5 overexpression systems, and we didn't really see a really strong phenotype in that system. What you can see here, though, is that when we have ILC2s introduced to the system, we start to now increase our recruitment of eosinophils. And so we thought that one of the cytokines was probably mediating that mechanism. If we look at the frequency of cells that are being recruited into the tumours versus tumour weight, so eosinophil recruitment, we find that there's a very strong relationship between the recruitment of eosinophils and the size of the tumours. And if we now look at the system where we delete raw alpha, so delete some of the ILC2s, we now find that there's a bit of a fairly strong reduction in the number of eosinophils, although eosinophils haven't stopped being recruited, it's just proportionately the number is significantly reduced. And so this sort of suggests that there's multiple mechanisms that are involved in recruiting eosinophils to the tumour. In this situation, we then wanted to ask, well, is the GM-CSF producing ILC2s? Is that really part of the mechanism, or is it the IL-5 that could be driving this response? And so if we take our system, now this is a RAG2 gamma-C mouse without any transfers, we have about 40% of our cells are recruited are neutrophils, and just a couple of percent are eosinophils. We then look at those that are transferred with ILC2s from wild-type mice, and we now have a much larger percentage of eosinophils that are recruited. And then if we recruit, if we introduce ILC2s that lack GM-CSF, we now have quite a significant expansion of eosinophils and neutrophils. And we were very interested, and this is shown by the frequencies of the cumulative data in the lower panel. And you can see that while the neutrophils are also increased, so too really are the ILC2s, and this is a very significant difference between the wild type and the CSF2 knockout mice. So how do we get a handle on what might be happening with the ILC2s in GM-CSF? And so we devised a system in which we're able to take our different types of cells, isolated from naive mice from the intestine, and then culture them with recombinant IL-7 and IL-33. And IL-33 is known to amplify the ILC2s. Do that in vitro, and then take some of that conditioned media from the ILC2s and apply that to eosinophils. And so then after 17 hours, we could isolate those eosinophils and do real-time PCR to try and understand what some of the changes might be for those eosinophils. And so we looked at a whole host of genes, and we were really facilitated in this by collaborating with Ariel Munis in Israel and his postdoc Sharon, who really had some really engaging conversations that I think would never have actually happened without COVID, and have resulted in a number of collaborations now. And so we pinpointed a number of genes such as GATA2, EPX, EU1 and EU2, which are essentially effector molecules in eosinophils. And you can see in the absence of GM-CSF, we fail to upregulate these sorts of genes. And so it appears that the GM-CSF is really driving it, inducing the effector function of the eosinophils that might be involved in the tumour. So do these eosinophils actually drive the anti-tumour response? And so again, we were very fortunate. We found a group who had a colony of mice that they were just about to discard, and these are eosinophil deficient mice, and they're colloquially known as the FIL mice. And so here you can see where we've introduced the ret tumour and then measured the tumour size and the overall survival of these mice. And you can see that there's quite a striking difference in the size of the tumours that develop, and here we've measured them at day 17. And this was quite statistically significant. Again, we saw that there was a change in the capacity of the animals to survive. So if you lack eosinophils in your entire hematopoietic compartment, regardless of the mechanism of recruitment, then you're at a much poorer situation for defending against the establishment of these melanoma tumours. If we look at the TCGA database, we find that the probability of having ILCs and eosinophils is directly related. And so here we analysed a much bigger database or sample size, so nearly 400, just over 350, nearly 400 samples. And then we're able to combine all of our data and ask, how does the ILC signature and eosinophil signature relate to each other? And overall, the ILC2 high, eosinophil high patients have a much better potential outcome or survival time compared to those that have low expression on either of those two parameters. And you can see that the survival time is doubled by having this high ILC2 signature combined with the eosinophil signature. So does a more specific ILC2 deletion actually impair the anti-tumours? And so this is a second model in which we have what we call the ICOS-2T mice, and these are mice, again, generated by Andrew Mackenzie. And so in these mice, these are conditional deletions. So the virtue of these mice is that you can induce a tumour in the mice, and it's in the context of it in a completely replete immune system, so containing both the innate and the adaptive immune system. And so now we're testing the ability to just deplete ILC2s in the context of that adaptive immune system. And so we treat the mice initially with tamoxifen because it's an inducible system, and then we're able to give them the ret tumour and then analyse the mice at various time points after that. And so this just shows you what the outcome of that type of experiment is. One thing we do is we treat with recombinant IL-33. This amplifies the ILC2s. It also increases the efficiency of the deletion in this model. And so we've compared B6 mice that are treated with PBS, those that have amplified ILC2s, with the notion that if we simply just amplify ILC2s, we can create a stronger response from these ILC2s. We have the control mice that have been given IL-33 to achieve a similar thing to the B6 mice, and then those mice in which we've amplified the ILC2s and then deleted them. And so what you can see is once you start deleting the ILC2s, you start to get a poorer response from these mice. And so it's this blue bar here that really reflects the loss of these ILC2s. And so we end up with this complete dysregulation. And if you simply amplify the ILC2s, then you actually reduce the rate of the development of these tumours. So it looks like we can manipulate the ILC2s to some extent. Could this effect actually be mediated by CD8 T cells? Because we know that they have very important effects. And so when we do the analysis of comparing the ILC2s to the tumour weight and comparing the frequency of CD8 T cells to the tumour weight, we find that the relationship actually exists amongst the ILC2 subset as opposed to the CD8 T cells. And so it's quite conceivable that ILC2s are doing something at this early stage in the anti-tumour effect, and that the functions of CD8 T cells are extremely important, but operating at a different time frame and using different mechanisms. So the question really is, why can't we do better in tackling a cure for melanoma? Why is it when we amplify the ILC2s that we don't get a complete remission of those tumour cells? Maybe the problem is just way more complex than that. But the other thing that we thought about is maybe we just don't have all the mechanisms that could be induced in this setting to allow us to do a better job of potential designs of immunotherapy. And so with that in mind, we went back to our single cell database and then started to look for a number of different genes that could be modulating the effects of ILC2s. And so you can see here that we've identified that quite distinct from some of the other cell types, is that ILC2s express high amounts of the inhibitory molecule PD-1. You can see this by FACS on the right-hand side in the BRAF mice. If we look on the human samples and do staining for ILC2s that are expressing PD-1, you can see that there's a small fraction of these cells occurring directly in the tissues, suggesting that maybe the brake is being put on these cells as they are being recruited into the tumor microenvironment. So these tumors actually express, these infiltrating ILC2s express high levels of PD-1, but if you look at a variety of different checkpoints, you find that there are a number of different checkpoints or inhibitory receptors that are being expressed by these ILC2s. And these are somewhat distinct from the checkpoints that are being expressed necessarily by different types of T cells or NK cells and gamma delta T cells. If we look at whether those ILCs are actually expressing just one type of inhibitory receptor, you find that in fact most of them are expressing multiple inhibitory receptors. So on the one hand, these are probably curtailing their immune effectiveness, or on the other hand, it potentially makes them candidates for modulation in immunotherapy. So the first one we really looked at, and the one I'm going to tell you about today, is PD-1 and what PD-1 is actually doing. And so we looked at PD-1 deficient mice, and so these are completely deficient in their hematopoietic system. And you can see here that when we do red inoculation, the tumor size is reduced in the absence of PD-1. And so somehow the expression of PD-1 is putting a brake on the immune system, dealing with the emergence of these tumor cells. If we then take these cells, similar to our previous adoptive transfer experiments, and transfer in PD-1 deficient cells, they have an effect very similar to the B6 mice. So that simply removing the inhibition of ILC2 almost completely restores the capacity of these mice to restrict the development of a tumor cell. And that's completely at odds with this RAG2 gamma C mouse that lacks both the ILCs and the adaptive immune cells. So what's going on here? So we looked deeper at what we were doing with our IL-33 stimulation. And so here you can see IL-33, this is in vitro cultures, is upregulating effector molecule KLRG1 in our ILC2s. And this occurs between day 2 and day 5, you get this massive upregulation. What also happens is you increase the capacity to produce cytokines, so both IL-5 and GM-CSF. And so these are all positive outcomes from our stimulation. But at the same time, we're doing something very negative to the cells. So we're inducing the expression of PD-1. So while we're driving forward, we're also putting the brake on simultaneously. So what does this mean? This probably means, at least in our mind, was that perhaps when we're driving through that IL-33 stimulation, the activation and proliferation of those cells, we're driving the expression of PD-1. If we could block the expression of PD-1, we could get better outcomes. And so this experiment shows you that, where we've taken these mice, the mice, and then we've treated them with anti-PD-1. We've treated them with the IL-33 or IL-33 and anti-PD-1. And so in the control mice, you can see the tumors develop. But once we start treating with both anti-PD-1 and IL-33, we start to have fairly important impacts on the development of the tumor. And so what's happening here? So here you can see the control mice, those treated with anti-PD-1, with the IL-33, and then the combined therapy. So we're actually driving this enormous proliferation of the cells. We're driving through the IL-33, the production of these effector type ILC2 cells. And with the anti-PD-1 and the IL-33, we're not restricting the development of those effector cells. So presumably, we're actually allowing those arm cells to be functional. And so here you can just see expressed as a different manner. And so that the number of these effector ILC2s per gram of tissue is actually significantly increased. And this occurs both in the spleen and systemically, which is to be expected since we're giving the treatment systemically. So the question is, are we changing some of the other cells that might be involved in this process through this combined therapy? Or what are we doing in that combined therapy? And so here you can see what we've done is really try to map the development or the recruitment of eosinophils into the tumour microenvironment. And so in our anti-PD-1 treatment, we've got a small number of eosinophils that's just a little bit different to the control mice. When we put in IL-33, we increase that recruitment of the eosinophils. And then when we put in anti-PD-1 and IL-33, we significantly increase the number of eosinophils that are coming into this system. And again, this is shown as per gram of tissue that were recruited into the system. And you can see at the tumour site, and not so much at the in the spleen, this combined treatment is really recruiting large numbers of eosinophils into the tumour itself. And so we think that this recruitment of the eosinophils into the tumour has profound effects in being able to restrict the development of those tumour cells. So just to provide a summary of what we've described is that when we have a melanoma, primary melanoma tumour cell, we get this very rapid early influx of these ILC2s. And these ILC2s can mediate anti-tumour responses principally through the cytokines such as IL-5 and GM-CSF expression. And this really drives the recruitment of eosinophils into the tumour microenvironment. The GM-CSF is increasing these effector molecules in the eosinophils. And while this is happening, we have the slow but progressive induction of PD-1, not dissimilar to what we see in T cells that become dysregulated in the tumour microenvironment. This in itself inhibits any of the positive effects by the inflammatory response of driving that recruitment of ILC2s. However, we can overcome this by putting in IL-33 to amplify these ILCs and simultaneously blocking the effect of these inhibitory receptors. And in our case, we've tested PD-1, but it's likely that there's a whole raft of different inhibitory receptors that could be synergistic with T cells. And by combined immunotherapy, we can improve anti-tumour responses. And so what we hope that we've been able to demonstrate is that perhaps this is a bit of a game changer. We can change the balance of protection by modulating what's happening for the innate immune system and the adaptive immune system. And similar to the innate immune system, these are similar to the adaptive immune system, these innate cells are actually expressing a raft of different receptors that could be co-opted in immunotherapeutic approaches. So in our case, we've got a PD-1 expressed by these ILC2s. And even in the case of PD-1 on the ILC2s, we get this dysregulation of the ILC2s. So if we think of it temporally, we've got ILC2s coming into the melanoma microenvironment, and they're being sequentially dysregulated. We then have T cells come in a little bit later, and they also become dysregulated and unresponsive. So we have this really stratified way in which cells are coming in, but also being dysregulated by the actual impact of the tumour microenvironment. However, one possible approach is to think about these ILCs as being the early responders and coming into the situation much, much earlier than we would normally think of a T cell coming in. And certainly a well-formed adaptive T cell that might have any significant memory type features about them. And that means that we could target this much, much earlier than perhaps we might think about targeting for T cells. It also means that these will probably be the first cells that are entering a tumour microenvironment when we have metastatic lesions. And so while this might be quite hard to target for a primary lesion because we're unaware of those ILC2s coming in, patients are likely to be asymptomatic, we will know after the primary diagnosis that metastases are likely to happen. And therefore, we could start targeting this early ILC phase well before we see the consequences of dysregulation of T cells. So with that, I'd like to thank the people in the lab who've been absolutely instrumental in driving this work. As I've mentioned, Nicholas Jacquelot really has pioneered this work and deserves full credit for making this such a success. But he's been also ably helped by Cyril Cilliard, Angela Wang, Mary Camilleri, and Kylie Leong, together with other people in the lab who've helped in various ways. We've had a number of collaborators at the WeHi who've assisted us in very kindly providing access to mice, some of which before they were published. We have a number of collaborators, so they live in Newton John Cancer Centre and at the Peter MacCallum Cancer Centre, who've really made a pathway forward for us to access human tissues, which initially was very difficult to be able to drive that forward. We've really enjoyed the collaboration with Ariel and Sharon in trying to understand a little bit more about eosinophils and hope that those collaborations will continue to move forward. And there's a whole raft of other people who've been involved in this particular project and have really made what was a very difficult problem right at the beginning a very enjoyable journey and has shown us a number of mechanisms that I think at least our lab hadn't really understood how complex this interplay was and the number of features of an immune response that we have to consider if we're to make inroads into what's happening in an immune response. So thank you very much, everyone.
Thank you very much, Gabrielle, for this really fascinating talk. I see, okay, thank somebody like Elena shared this slide. So, so before that closing, I just wanted to highlight that you can ask a Q&A question to Gabrielle via Twitter. First, you have to search for the account, the Global Immunotalk. Second, you have to find a tweet that will say ask question for Dr. Gabrielle Belz here. And after reply to the tweet with your question and mention at Global Immuno speaker. And on this, I will again close the meeting and thank you very much, Gabrielle, for joining. Thank you. Bye-bye.
Thank you very much.